Okay, here we go. All right, so what we're talking about today, the focus of today's lecture, is talking about genes and the basics of kind of genetics. So this lecture is intended to make it so that students who don't have a very solid biology background, because people are coming from all kinds of different backgrounds when they show up here, we want to make sure that everybody's on the same page, that everybody has the same basic knowledge about genes and DNA, so that we can start to talk about how that relates to our psychology and how that relates to how our brains are built, and therefore our psychology. So we're going to kind of just lay that down tonight, and then we'll start to really get into more of the details of how it applies to biopsychology specifically in the next lecture. So I'm going to start with talking about chromosomes, what they are, how we get them, a little bit of the background. Okay, so chromosomes are in every cell of your body. Every cell of the human body has a nucleus, including neurons and heart cells and lung cells and all the different kinds of cells have a nucleus. And in this nucleus, there are chromosomes. There are, in fact, in every human cell, 23 pairs of chromosomes. So if you have 23 pairs of chromosomes, how many total chromosomes is that?
46.
46. Awesome. Whether you did that by addition or multiplication, you all arrived at the right answer. Great job. Okay. 22 of those pairs are called autosomes. There are 22 pairs of autosomes, so how many total autosomes? 44 in every human body cell, whether you're talking about a neuron or a blood cell or whatever it is. And then there's one pair that are called sex chromosomes. In most people, if you have an X and a Y chromosome, you'll be genetically male. And in most people, if you have an X and an X sex chromosome, you'll be genetically female. However, there are exceptions to this rule. There are people who have two X's and a Y, or an X and two Y's, or other variations. But most people are either XX or XY with their sex chromosomes. You know that your genes influence your body and your physical makeup. You know that you got genes from your mom and genes from your dad, and you can see some of those features in yourself. Your eye color, or your skin color, or your hair color, or how tall you are. All of those different things, all of those different variations, right? Some of that has to do with differences in your genes. Some of that has to do with things that you inherited from your parents. The only cells in the human body that don't have 23 pairs of chromosomes are your gametes. Your gametes are your sex cells, your eggs, or your sperm. Eggs and sperm have exactly half as many chromosomes as all of the other body cells. So all of your body cells that make up you have all of your chromosomes. But when you're going to pass on your genes to your child, you give half. And the other person, who's the other member of the parent team, gives the other half. It wouldn't make any sense for you to give all of your DNA and your mate to give all of their DNA, because that would be 2 whole people, right? So every person is half mom and half dad. Every person got half of their genes from their mom and half of their genes from their dad. In other words, if you were to look at a sex cell, if you were to look at a gamete, it wouldn't have 23 pairs of chromosomes, it would just have 23. So the 23 that goes into one egg and the 23 that goes into one sperm, they come together now to make 23 pairs in that new person. Does this make sense? Pretty straightforward? Okay. So, let's imagine that you were inheriting, let's say we're creating a new human, okay? We're putting an egg together with a sperm. When they first come together, you're going to get one number 1 from mom and one number 1 from dad. One number 2 from mom and one number 2 from dad. One number 3 from mom and one number 3 from dad. And so on and so forth for all of the sex cell, for all of the chromosomes. Make sense? Okay? So far, so good. The substance that makes up chromosomes is called chromatin. You can see it right here on the slide, chromatin. Different kinds of chromatin, heterochromatin, euchromatin, but it's all chromatin. And then there are some other proteins that are in there that hold the structure of the chromosome together, but those aren't part of the DNA. The important part of the chromosome, the part that we care about, the part that we need, the part that is the subject of this lecture, is the DNA. Are the instructions that are carried on the chromosomes. That's what DNA is. DNA is a set of instructions for making you. DNA is a blueprint that contains all of the directions for making all of the parts of your body. So, when you were first conceived, when sperm first met egg, there was an egg that had 23 chromosomes in it, and there was a sperm that had 23 chromosomes in it. They came together and made you, and you have 46 chromosomes in you. This cell, when you were first conceived, is a stem cell. It hasn't turned into any specific kind of cell yet. Remember that from last time? Remember stem cells when we talked about that? Okay, so it hasn't turned into any kind of cell yet. So when you were first conceived, the first thing we need to do is just make more. Make more cells. Replicate, 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 replicate. More and more cells. And then later in development, they turn into what they're going to be. A neuron turns into a neuron, and a blood cell turns into a blood cell, and a lung cell turns into a lung cell, and a bone cell turns into a bone cell, and so on. But the directions for making any of those things are all contained in all of the cells. All of the cells have all of the instructions for being any kind of cell. All the DNA is in every cell. What's different in different cells is which genes are expressed. Which pieces of the DNA are expressed. In neurons, you remember neurons? Right? We've talked a lot about neurons. Please tell me you remember neurons. Okay. You have to have all the instructions in a neuron for making a neuron. You have to have the instructions for making dendrites. You have to have the instructions for making receptors. You have to have the instructions for making myelin. You have to have the instructions for making neurotransmitters. You have to have the instructions for making microtubules. You have to have all those instructions. And those all get turned on in a neuron, and it makes a neuron become a neuron. And that's a different kind of cell than a blood cell. A blood cell has the same DNA in it, the same nucleus with the same DNA in it, but different parts of it are active that make it a blood cell. Okay, we are going to come back to this. We're going to revisit this a lot. But you've heard me mention the word DNA, the word genes. That's what makes up a chromosome. That's the important stuff in the chromosome, and that's the stuff that we're talking about here. The genes. If you were to, so this is an electron microscope picture of the sex chromosomes, right? An X over here and a Y over here. So this is a male person, okay, male human. If we were to take a picture and draw it, you can see what's actually in here up close. So if you were to unwind it and look at it unfurled, you can see the DNA structure. And it's called a double helix. A double helix is like a ladder that's in spirals, that's spiraling around. And that's how our DNA is put together. Mitosis is the process of making whole new cells. So during development, when you're making new stem cells, you're making new stem cells through mitosis. And then, as development goes on, you're making other cells through mitosis. And you're making new cells through mitosis right now. You're making new skin cells, you're making new muscle cells. Any cell that's wearing out, any cell that you need to replace, you're doing mitosis to create copies of the cell. You need to build more of that thing that you're made of. You need to build more. Let's say, you worked out really hard and you really tore up your muscles, by working out and lifting weights. Now your body needs to go in and repair those muscles. Well, you make new muscle cells through mitosis. And so on and so forth. Neurons are often hard to replace, or generally speaking, hard to replace in your central nervous system, but elsewhere we do replace them, usually. So I'm going to talk about the steps of mitosis. The steps that we have to take in order to make a whole new cell. And the reason that I have to talk about this is so that we can get to the next thing and the next thing and the next thing. So you got to follow along with the first most basic thing so that we can get to the next steps, okay? So let's just start with the phases of mitosis and the phases of a cell's life in general. Most of the life of a cell is spent in interphase. Interphase is where a cell is just being a cell. It's just being a skin cell, or it's just being a heart cell. It's doing all of its cellular functions. It's doing all of the things that that cell needs to do at that stage. But sometimes we need to make a new cell, and that's what's happening in mitosis. So there's a phase of the cell's life where it doubles all of its chromosomes. It replicates all 46 chromosomes. That phase is called S phase. Okay, in S phase, you're synthesizing a whole new set of chromosomes. Because the point is you want to make two cells. You want to make a new cell. So you have to copy everything. If I'm a skin cell and I want to make a new skin cell, I have to make a copy of all of me to make a new skin cell. During prophase, that's the first stage of mitosis. Prophase is the first stage of mitosis. In prophase, you have those 92 chromosomes now, because they doubled, they replicated themselves. The DNA completely replicated itself. Yes?
So S phase happens between interphase and prophase?
Yes. It's a part of interphase. S phase is a part of interphase. Good question. Okay. So during prophase, now you've got these 92 chromosomes. Enough to make two complete new cells. But we need to do that. We need to separate the cells into two completely new cells. So in prophase, first, those 92 chromosomes, the first thing they do is they kind of untangle themselves. At first, they're all just kind of balled up in the nucleus of the cell, and they kind of have to untangle themselves to become discrete chromosomes. They make themselves into their discrete, instead of just being a big jumbled up mess in the nucleus, they separate themselves out into the distinct chromosomes that you see, like on a karyotype. That's a karyotype. I'm going to talk about that more in a second. Okay. So now you've got the chromosomes doubled to 92, and you've got them all nice and discrete. Now we have to start separating them apart. And during prometaphase, which at first they actually didn't have a prometaphase, they put it in because they realized it would be clearer if they made one more step. The nucleus that's been surrounding all of the chromosomes dissolves. And microtubules that are in the cell invade the space. Microtubules come in where that nucleus used to be, and they attach themselves to the centers of each of the chromosomes. They attach themselves right at that centromere, that center part of the chromosome. The ends of the chromosome are called the telomere, which you see in this slide, telomere, or telomere. I've heard people say telomere. That's fine too. Centromere is right here in the middle. And that's where the microtubules insert themselves. The microtubules go through the centromere. And then during metaphase, they all line up. During metaphase, all the chromosomes attached to the microtubules all line up at the very center of the cell. And that's so that the microtubules can pull them apart. Because in the next phase, anaphase, the chromosomes are going to be pulled apart. 2 of the number 1s are going to go in one direction, and 2 of the number 2s are going to go in the other direction. 2 number 2s in one direction, 2 number 2s in the other direction. 2 number 3s in one direction, 2 number 3s in the other direction. All of the different pairs are being pulled apart. In telophase, or telophase, however you want to say it, that's the last step of actual mitosis. At the end of mitosis, in telophase or telophase, the microtubules go away, they disappear, and the process of cell division begins. The 2 now complete nuclei are at the 2 ends of what used to be 1 cell, but is about to be 2 cells. So you've got 46 chromosomes over here, and 46 chromosomes over here, and a new nucleus forms around these 46 chromosomes, and another new nucleus forms around the other 46 chromosomes, and that's the end of mitosis. There is one more step that we call cytokinesis that's technically right after the end of mitosis, but cytokinesis is where a ring forms around the center and it pinches the 2 cells off from one another. A ring comes in and starts to squeeze down, 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 down until it pinches the 2 cells apart from one another. And now you have 2 fully complete cells. And we're going to watch a video that shows this process happening, because sometimes it can seem a little bit obscure, but then when you see it in motion, it makes more sense. So we're just going to watch the first 2 minutes or so of this video. I hope I don't blow your ears out, guys. I think we should be okay.
Cell division is required for an organism to grow, mature, and maintain tissues.
You know what? I'm going to turn it up just so it's nice and loud on the video.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes metaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the 2 sets of chromosomes. Mitosis is now complete. After mitosis, 2 new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of 3 stages called G1, S, and G2. G1, or gap1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first...
Okay. Okay, so the way that we get these nice karyotypes, like let's say that you were going in to get genetic counseling or something before you got pregnant. They'll show you your karyotype, and your karyotype is a picture of all of your chromosomes. And the way that they show you that is by starting mitosis. They take some of your cells and they start to put them through mitosis in a substance that encourages cell division in the lab. And as that process begins, as mitosis begins, they stop it during metaphase. And they have all these nice, distinct chromosomes now. And so they take them and they separate them out, in the petri dish. And then they can take a picture of it. And they can cut them out and line them up. Like they'll line the 2 number 1s up together and the 2 number 2s up together and the 2 number 3s up together so that you get this nice picture. Meiosis. Okay, so we've been talking about mitosis. The process of creating a whole new cell, a whole new piece of you, a whole new cell of you. For meiosis, I've heard people say meiosis, that's fine too. For meiosis, you just need to make a sperm or an egg. And like I said at the beginning of class, a sperm or an egg only has to have half as much DNA as the rest of you. Because you need that half to come together with the half from another person in order to make a whole new human, right? So here's the thing about that though. When your chromosomes are in you, in one of your cells, when you were first conceived, when the egg first came together with the sperm, one of the chromosomes was from mom and one of the chromosomes was from dad for each of the pairs, right? Half of your genes came from your mom and half of your genes came from your dad. So let's say these are the two members of the number one pair. Here's one and here's the other one. Right? So this would be like the number one pair. But this could apply to any of them, okay? But let's just say it's the number one pair. So this is A and B of the number one pair. When they're inside you, they mix themselves up with each other. So part of this chromosome will come off and exchange places with the other part of the other chromosome. And so on and so forth for the entire chromosome. The different pieces will trade between the two members of the pair. The two number ones will trade back and forth pieces. The two number twos will trade back and forth pieces. The two number threes will trade back and forth pieces, and so on and so forth, for all of the members of the pair. It doesn't matter because it's all you. But when you go to make a gamete, when you go to make an egg or a sperm, you give one member of the pair to one egg, and the other member of the pair to the other egg. Or if you're making sperm, you give one member of the pair to one sperm, and the other member of the pair to the other sperm. But they're not like when you first got them. You don't have all grandma over here and all grandpa over here. They mixed up while they were in you. They exchanged pieces while they were in you. And that's why your children, when you make an egg or a sperm, doesn't have all grandma or all grandpa's genes. They have some of both. Because the two chromosomes have been switching back and forth their pieces. And so then when you go to make an egg or a sperm, you don't have two identical cells, you have two different cells. Instead of it producing all of the copies of all of your DNA and making an identical replicate, okay, so you're making another piece of you in mitosis. You have one cell that has all your DNA in it, and you need to make another cell that has all your DNA in it. It's all you. But when you're going to make an egg or a sperm, you have to just give half of your DNA. But you don't give all of your mom's genes or all of your dad's genes. You give a combination of both. And the reason that you do that is because of this swapping back and forth of the parts during meiosis. So meiosis has all of the same basic stages as mitosis. You're going to see the chromosomes lining up and the microtubules invading the space and the microtubules pulling the chromosomes apart, and half of them go to one end and half of them go to the other end. But this time, instead of it being 92 chromosomes that are separating into two sets of 46, instead you have 46 chromosomes that are separating into two sets of 23. So that this set of 23 can someday meet another set of 23 from some other person and make a whole new person. It would be nice, I guess, in some ways, if we could just be like some plants and other organisms that do asexual reproduction. So I have a plant, I have a golden pothos on my refrigerator. And I've had her since I was 17, I think, 17 or 18. And she has spawned hundreds of offspring. She's huge. She's spawned hundreds of offspring because all you have to do is cut off one of her leaves, stick it in water, and it grows a whole new plant. It grows roots and it'll grow a whole new plant. So her daughters are exactly identical to her. But our children are not identical to us or to their grandparents. They're their own set of genes. And the reason for that is because that's how meiosis occurs. If you're a woman who has eggs, all of that happened before you were even born. All of your eggs are fully developed by the time you're born. I mean, they're all created. They haven't matured yet, but they're all in there. They're all made. Sperm get made throughout the man's life. So he's doing meiosis all the time. This idea that chromosomes during meiosis, that when chromosomes are making gametes, have non-identical daughter cells, that the two cells that eventually get produced at the end of meiosis are different from each other. This one has this piece that's like grandma, and this one has this piece that's like grandma, or grandpa. This one has this piece that's like grandpa, but this one has this piece like grandma, and so on and so forth. They're all mixed up. And that is why our children have usually, generally speaking, have some kind of combination of both grandma and grandpa's genes because of this mixing up process. And that's called the law of segregation, or Mendel's first law. This idea that chromosomes separate when gametes form, and the gametes form non-identical daughter cells, that's the law of segregation. The guy who discovered this was named Mendel, and so the law of segregation is called Mendel's first law. Mendel was a monk, and he conducted his studies on pea plants. And he was very methodical. He recorded everything about the plants. He recorded the colors of the flowers. He recorded whether the pea pods were wrinkled or smooth. He recorded all of this information about the different plants. And then he bred different plants together to see how the baby plants would turn out. Some plants that have flowers, use sexual reproduction. My golden pothos doesn't have flowers. It uses asexual reproduction. But some plants, a lot of plants have flowers and they have plant sex. You know, the birds and the bees, right? And so they follow the same basic concept of making a new plant as you do to make a new person. And so he figured all of this out by studying all of these different characteristics in the different plants. Okay, we're going to come back to that. We're going to come back to that. But I want to talk about other stuff that genes do. Okay? So far we know that genes do two different things. You can reproduce an entire cell and make a new you, a new piece of you, a new cell that's you. That's mitosis. Or genes can separate out half and half, and that's meiosis, to make a gamete, to make an egg or a sperm. But cells also have to maintain themselves. So for instance, let's say you have a neuron, a neural cell, and you are taking an antidepressant. Remember that last time? You're taking an antidepressant. Or let's say you're learning Remember, you guys just learned about learning how you're building new AMPA receptors? The instructions for building the AMPA receptor is in your DNA. The instructions for building the serotonin reuptake transporter is in your DNA. The instructions for building every receptor, every neurotransmitter, every microtubule, every part of that cell, and it's so complicated, right? Just a neuron is so complicated. It has all these different parts. And we only talk about a few of them in this class. But your DNA has the instructions for making all those parts. Your DNA is a set of instructions for making proteins. One gene makes one protein. A gene is a unit of DNA that is the instructions for building a protein. So you have a serotonin reuptake channel gene. And that gene is a set of instructions for building the protein that is your serotonin reuptake channel. And you have genes that say how to build an AMPA receptor. The gene is a set of instructions that says if you put together things like this, it will make an AMPA receptor. So when that second message comes to the cell that says it's time to make an AMPA receptor, this person is learning in class today, yay, even though it's Tuesday night, okay, this person is still learning something in class. That AMPA receptor is being built because your DNA is being signaled to make one. The second messenger says, hey DNA, build a new AMPA receptor. DNA goes, okay, here's the gene for that. Those instructions are read, and it creates the protein that makes that serotonin reuptake transporter. And that's true for every piece of your entire body. Everything in your body, all of those tiny details that are in every cell, all of those instructions, every one is a gene in your DNA. That's why chromosomes are so huge. Because think about how much there is to you. Every cell is built differently. And it's not just how you build the cell. There's also instructions for getting those cells to where they're supposed to be during development. So we have instructions for all of them. Instructions for the glial cells. Instructions for all the pieces of the glial cells. That's what genes are. Codes for making proteins. If you were to read a little segment of DNA and follow its directions, it's going to tell you how to make a protein. So let's say that we're talking about the color of a flower. Or, in a human being, let's say we're talking about the color of your eyes. There's a gene, or maybe a few genes, that determine that. In the case of the plants that Mendel was working with, it was one gene. There was one gene at a certain place on one of the chromosomes that said, make the flowers this color. That gene, that place on the chromosome, is called a locus. Locus is the Latin word, and loci is plural. Locus is the Latin word for place. It just means the place on the chromosome. So let's say that the gene for flower color is at this place, this locus on the chromosome. It will be at this locus on both members of the chromosome pair. So the locus for flower color is here on A1, and it's also here on B1. Both members of the chromosome pair have the same locus. They match. They code for the same thing. They code for flower color. Or let's say eye color, or skin color, or serotonin transporter. So you got instructions from your mom for making a serotonin transporter, but you also got instructions from your dad for making a serotonin transporter. That's the locus at both chromosomes. However, the instructions you got from your mom are not necessarily the same as the instructions you got from your dad. So with Mendel's flowers, he had some flowers that were white and some flowers that were purple. So the place where color of flowers is on the chromosome is the same locus, the same place. But some of the instructions at that locus say make white flowers, and some of the instructions at that locus say make pink flowers. They don't necessarily have to have the same instructions. It's for the same thing, but it might be two different types of that thing. So it's both there. It's the locus for eye color, but it might be two different eye colors. Right? My mom had blue eyes. My dad had brown eyes. I have brown eyes like my dad. But my mom gave me her genes too. You just don't see them. But they're still in me, right? You see my dad's eye color. Those two different variations in eye color, or the two different variations in flower color, the variations are called alleles. So there's a brown-eyed allele and there's a blue-eyed allele for eye color. And Mendel, like I said, did this with pea plants. But the example that I'm going to give you to illustrate how this works is eye color in humans. Now, the first thing I want to say is that eye color is actually more complicated than this. Because people always want to ask questions about, well, what if you have an occlusion in your eye? Or what if your eyes are blue-green? Or all of these different things. Okay. In truth, there's more than one gene that determines your eye color. And there are other factors from the environment that can influence your eye color too. But the inheritance pattern often works this way, and it's easy to understand. So I'm going to use this as an example. Okay. Look at this lovely couple. They both have brown eyes. However, I happen to know that woman. Eye color often follows a pattern of inheritance that Mendel looked at in his pea plants too. And that pattern of inheritance is called a dominant recessive relationship. Okay. Brown eyes, in this simplified explanation, brown eyes are dominant. Meaning, if you have a gene for brown eyes, you will have brown eyes. It doesn't matter what the other gene is. The other gene can be another dominant one for brown eyes, and you'll have brown eyes, or it can be a recessive one. And recessive genes are like for blue eyes. Blue-eyed genes are recessive. Meaning, in order for you to have blue eyes, you have to have two blue-eyed alleles. If one of your alleles is brown, you're going to have brown eyes. So in order for you to have blue eyes, you have to have 2 recessive blue-eyed alleles. My mom had blue eyes, so I know she had 2 recessive blue-eyed genes. So she could only pass on to me a blue-eyed gene. That's all she had. And so I know that I have 1 blue-eyed gene in myself. I know that even though you see brown eyes when you look at me, that brown-eyed gene came from my dad. My mom gave me a blue-eyed gene. That's all she could give me. That's all she had. So I know that I have that in there. That, the way my alleles are set up, is called my genotype. The thing that you see in my appearance, the brown eyes, that's my phenotype. Phenotype is the observable outcome of the genotype. The genotype says one thing, but the phenotype is what you see. Whether you're talking about in my eye color, or whether you're talking about in my serotonin transporters, or any other thing. The genotype is what you can measure. That my eyes, or the phenotype is what you can measure, that my eyes are brown. The genotype is what's in my genes. And you wouldn't know it by looking at me. But I know it because I knew my mom and what color eyes she has. Now David, we don't know. Okay? Or I mean this handsome guy. David has brown eyes, but both of his parents also have brown eyes. That doesn't mean that he doesn't have a recessive gene in there. Now, okay, David's Mexican-American, and I think my Mexican-American peeps can back me up on this. Every once in a while, you have a family member who has blonde hair and blue eyes. Yes, thank you. Everybody's nodding their head, okay? Because there are recessive genes in there. So let's just imagine that David's genotype is the same as mine. We can never be sure what his genotype is, but let's just, unless we did genetic, tests on him, but let's just say it's like mine for the sake of argument. What that means is that if we decide to create offspring together, and we did, this is how we can pass on our genes. Okay, this is me, and this is Dad, David. I can pass on my dominant brown gene, and he can pass on his dominant brown gene. I can pass on my recessive blue allele, and he can pass on his dominant brown allele. Or I can pass on my dominant brown allele, and he passes on his recessive blue allele. Or we could both pass on our recessive blue allele. That means that in the case of all 3 of these people, 75% of our offspring will have brown eyes. This person has 2 dominant brown-eyed genes, and so they're going to have brown eyes. This person is like me, 1 dominant and 1 recessive, and so is this person. But this person got the 2 recessives, David's recessive and my recessive. And so 25% of the time, we will have kids that have blue eyes. The genotypes then can be 2 dominants, a dominant and a recessive, or 2 recessives. 3 different genotypes, right? You get this 25% of the time, this 50% of the time, and this 25% of the time, because these 2 are the same, right? So you have 3 different genotypes that we could make. But only 2 phenotypes, because these 2 genotypes both make the same phenotype. These 2 genotypes both make brown-eyed people. And as you can see, our darling daughter, Avalon, Biddy, as I talk about her, has brown eyes. But I have 2 other kids. and they have a different dad. Their dad is this guy, Randall. Great guy. One of my best friends in the entire world. Right? Totally your best friend too. Yeah. Okay. We have a weird family. Anyway, Randall has blue eyes. So, Randall can only make blue-eyed sperm. He has two blue-eyed alleles. Why? Because he has blue eyes. I know it's recessive. So he must have two blue-eyed alleles. So that means I can pass on my dominant allele 50% of the time. I can pass on my recessive allele 50% of the time. But he can only pass on the recessive allele. That's all he has. This state, when you have this kind of genotype like I do, it's called being heterozygous for that trait. Two different kinds. Hetero means differing. Heterozygous means I have two different alleles for that gene. This, where they're both the same, is called homozygous. And in this case, homozygous recessive. If you had two dominant alleles, you would be homozygous dominant. So as you can see right here, 50% of the time, my kids with Randall should have a genotype that's heterozygous, which means their phenotype will be what? Brown eyes or blue eyes? Brown. But 50% of the time, they're going to be homozygous recessive in their genotype. So what will their phenotype be? Blue eyes. And if you look at my two daughters, Erica has brown eyes and Angelica has blue eyes. Exactly what this kind of dominant recessive inheritance pattern would predict. So this law of segregation that we talk about with Mendel is based on the fact that there are two alleles at every locus, and they don't necessarily have to be the same. For a lot of things they are. Okay, so for example, almost all of us have DNA that says we should have 5 fingers on each hand. Almost every person's DNA says you should have 5 fingers on each hand. So whether that locus was your dad's or your mom's, they're identical. Whether the allele came from mom or dad, it probably says you should have 5 fingers. And that's true for lots of your characteristics. So for example, having 2 arms and 2 legs. Almost everybody has the same DNA. It says make 2 arms, make 2 legs. Make 2 eyes, make 1 nose, make 1 mouth, make 1 heart. All of those instructions that are almost always the same between people, the 2 alleles of whether you get it from mom or dad are the same. There's only 1 set of instructions, generally speaking, for lots of things. But then for other things, there's different ones. For eye color, there's all kinds of different alleles. You can have blue, or you can have green. Green is also recessive. So if you got 1 blue eye gene and 1 green eye gene, your mom had blue eyes, your dad had green eyes, you're going to have blue-green eyes. They will co-express. The same is true if you have 2 dominant alleles that are different. They'll co-express. Okay. So Mendel did this with his pea plants. He figured out by keeping track, he would say, okay, I have this plant and it's got green pea pod. I have this plant and it has a yellow pea pod. I breed them together hundreds and hundreds and hundreds of times. How do their offspring turn out? Well, half of them are green and half of them are yellow. And then I do that with yellow ones, and I do that with green ones, and I breed green with green and green with yellow, and I keep recording all of this. And that's how he figured out the dominant recessive relationship. just by keeping meticulous records over time. And then not a lot of people knew about it. He was a monk, so like, and it was at a time when, you know, there was no internet. So like Darwin didn't know any of this. Darwin, who was developing the theory of evolution by natural selection, he didn't even know about genetics. Mendel knew that. Ay, ay, ay. But these things are called Punnett squares, okay? Punnett squares are how he figured this out, and so you see these all the time in, you know, when we're trying to calculate dominance relationships, dominant recessive relationships. Okay. Boy, it got really far. Okay, cool. Is all of this clear so far? Now, if you have a background in biology, some of this is going to be really familiar. I mean, ideally, you started learning about this stuff in like elementary school, and then it got more and more reinforced as you went on. But I know, you know, not everybody gets a standard education, so we got to make sure we're all on the same page. There's SI for this, there's videos for this, okay, there's all kinds of resources for this, but you need to understand it so that we can move on to the next thing. So please ask questions if you have them now, or, you know, if you feel you need to reinforce this, do that until it clicks for you. Okay. This dominant recessive inheritance pattern explains a lot of diseases. So for example, cystic fibrosis, sickle cell anemia, and we'll talk about these as examples later on in the class. Tay-Sachs disease. But I do want to also point out that sometimes, no matter what your genes are, things can happen that prevent them from being expressed. So for example, you might have genes like this that code for a dominant brown-eyed allele and a recessive blue-eyed allele, but something happens during development that silences the brown one. And so all you see is the blue one. Things like that can happen, though they're rare. So this is showing the same thing as the brown-eyed, blue-eyed thing. It's just another diagram showing the same thing as the brown-eyed, blue-eyed thing. This is like David with one dominant brown-eyed allele and one recessive, and this is like me with one dominant brown-eyed allele and one recessive. So 3 times, 75% of the time, 3 out of 4 times, we will have a child with brown eyes. But 2 of those 4 times, they will be a carrier for the blue-eyed gene. And 1/4 of the time, they come out with the 2 blue-eyed genes, the 2 blue-eyed alleles, and therefore have blue eyes. And that's true also for like cystic fibrosis. If you have one parent, cystic fibrosis is recessive. So if you have one parent doesn't have cystic fibrosis, their phenotype is normal. And they meet up with another person whose phenotype is also normal. Neither of these people has cystic fibrosis because it's recessive. However, they both have the recessive cystic fibrosis gene. So 25% of the time, if those 2 people reproduce, they're going to make an affected baby, a baby that has cystic fibrosis. 50% of the times they're going to make carriers like themselves, and 25% of the time they'll make somebody who's homozygous dominant, and therefore neither has the disease nor is a carrier. Does that make sense? How you can be a carrier for a disorder too? In the same way as I'm a carrier for blue-eyed genes, I might also be a carrier for cystic fibrosis. But I wouldn't know because I don't have cystic fibrosis. I have one of the dominant genes, and I don't know what the other one is. I've never had genetic testing. But for like Tay-Sachs disease, there are certain ethnic communities that are more predisposed to Tay-Sachs, and so they will go in if they marry another member of their ethnic community, and they will go get tested because they're more likely to be carriers for Tay-Sachs. So they go in and they want to say, hey, I want to know if I'm a carrier, and I want to know if my spouse is a carrier, because that means 25% of the time we might end up with a kid who has cystic fibrosis. And that's something, I mean Tay-Sachs disease. And that's something you might want to know. Does that make sense? Okay, so far so good? Any questions about this good stuff? Carriers. Okay, cool. The law of independent assortment was the other of Mendel's laws. Okay, so two big Mendel laws. The law of segregation, which we already talked about. Then you're going to have the two daughter gametes that are different than the parent cell. They're not necessarily the same. They're not the same as each other, necessarily. But the law of independent assortment says that inheritance at one locus does not depend on inheritance at another locus. What does that mean in simple terms? It means that if you're looking at a set of chromosomes, and you're looking at the locus up here for eye color, that is totally separate than the locus down here that's for hair color. And that's totally separate from another locus at a completely different set of chromosomes for skin color. You know, so this is chromosome 1, this is chromosome, 16. And on chromosome 16, let's even say, okay, it's over here. This is for skin color. And so is this one and this one. Okay? So, you can get blue eyes and brown hair. Or brown eyes and blonde hair. Because those two things don't depend on each other. The different traits sort independently. A trait that's at one locus, like eye color, is not dependent on a trait passed at another locus, like hair color or skin color or whatever. So, when I am passing on my brown eye gene or my blue eye gene, that has nothing to do with whether I'm passing on my brown hair gene or I actually have a red hair gene because my mom had red hair. Okay? So, my two kids actually ended up, Erica has, usually if she's out in the sun, brown eyes and blonde hair. Angelica's the opposite. Angelica has brown hair and blue eyes. But that's not how it usually is. Usually you see brown hair or dark hair and dark eyes, and usually you see light hair and light eyes, but not always. And the reason that it's not always like that is because of the law of independent assortment. Those things come from two different loci, and so they don't have to be inherited together. Does that make sense? And the whole reason that this occurs, that you can make different eggs, some of my eggs have brown eyes with brown hair, and some of my eggs have blue eyes with red hair, and other of my eggs have brown eyes with red hair, and other of my eggs have blue eyes with brown hair. However, even though you might think, sometimes you might think by just looking at people casually that dark eyes always go with dark hair and light eyes always go with light hair. You might think that if you just looked at people casually. And so you might think that you always get light hair with light eyes and dark hair with dark eyes. But it's not like that. It can arrange any different way. You can get dark hair with dark eyes, or light hair with light eyes, or dark eyes with light hair, or light eyes with dark hair, or any combination, right? That's the law of independent assortment. And the reason that this occurs, like I say, is because before you make that sperm or egg, before you make it, before meiosis occurs, the two members of any chromosome pair switch information back and forth. They actually will swap their pieces. That is called crossing over and recombination. That is called crossing over and recombination. Because that's exactly what it is. The chromosomes are crossing over and recombining. The pieces of the chromosome are crossing over and recombining. And that's why you can get brothers and sisters that look nothing alike. Often they do, but sometimes they don't. And sometimes it's very striking. So for example, this is an interracial couple, and so they have different genes knocking around in there. And they had a set of twin daughters who are fraternal. They are not identical twins. And one of them just happened to get a lot of the dark genes, and the other one of them just happened to get a lot of the light genes. Now it could have been any combination. You could have got both of them with dark hair and dark eyes. You could have got both of them with light hair and light eyes. You could have got one with dark hair and light eyes and one with light hair and dark eyes. You could have gotten any combination. But statistically speaking, it's bound to happen somewhere along the way that one of the people is going to get a lot of the dark genes and one of the people is going to get a lot of the light genes. Statistically speaking, it's not very likely, but it's going to happen at some point, and those people are going to end up in the news because it's really interesting, right? It's very rare. And this couple, not only did they do that once, they did it twice. They had a second set of fraternal twins that were the same way. Beautiful. Awesome. Anyway, we have a video that's going to talk about this now.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for two genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now, consider the case where there is crossing over in the region between the two genes. Since the crossing over is only between two individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
Okay, so you hear this thing, linked genes. Linkage is a phenomenon in which it seems like the genes don't assort independently. Because a lot of the time you do see, most of the time you do see dark hair with dark eyes and light hair with light eyes, most of the time. Not all of the time, but most of the time. So why? There are even disorders that we've discovered where the gene for the disorder was because we knew about some other gene and where it was, and all of the people who had the disease had that other trait. So for example, you have people come in and they all have, let's call it disease X, okay, a made-up disease, disease X. And everybody who has disease X has oddly shaped ears, very large ears, okay? So you look where the gene is for ear size, and right next to it is the gene for that disorder, for that disease X. And so it seems like everybody who has these big ears also has disease X. That's called linkage. But here's the thing about linkage. It's a statistical phenomenon. It is not that genes are actually linked together by some like physical thing. It's only that they are close to one another on the chromosome. The closer they are to each other on the chromosome, the more likely they are to travel together. So if you have the locus for eye color right here, and the locus for hair color right next to it, most of the time, they'll go together. The only time that they sort independently is if the break, the crossing over and recombination, occurs exactly between the two of them. If a gene for eye color is up here, and the gene for hair color is down here, the chances that they will cross over and recombine are very high. Because crossing over and recombination can happen anywhere in between where one gene is and where the other gene is. Where one locus is and the other locus is. You can have crossing over and recombination happen anywhere in between them. So they will sort independently very frequently. Whereas if the genes are right next to each other, you would have to get a break, you would have to get crossing over and recombination right in between them for them to separate out and be inherited separately. So does it make sense that linkage is a statistical phenomenon determined by how close together two loci are for two genes, and not about them like being actually physically linked together? Does that make sense? Cool. Okay. And then I just have one little extra bit to say about this. Number one, all of these things that we've been talking about right now have been about dominant and recessive relationships and have been about a single gene responsible for a trait. Like there's a single locus for eye color, or a single locus for flower color, or a single locus for hair color. But the truth is, for most traits that you can see, they are not determined by a single locus. So for example, your skin color is not determined by a single locus. There are many loci that contribute to your skin color. That's why they're so all over the place. There are many loci that contribute to your IQ. Even though when you get your IQ score, it's one number. You know, your IQ is 112. That's one number. There are millions of your pieces of brain, millions of parts in your brain that are built by millions of different genes that contribute to your IQ. Height. Height has lots of different parts to it. Lots of different genes code for lots of different parts of your body and how fast you grow, things like that. And so you get a combination of genes. Like let's say you have a really tall dad and a really short mom. You're not going to inherit your tall dad's legs and your short mom's torso. It's not going to happen like that because a lot of genes contribute to your height and a lot of different factors. That is called a quantitative trait. A trait that many loci contribute to that trait is called a quantitative trait. You can also have co-dominant and co-recessive, as I've mentioned, and those kinds of things too. Okay. That's how we get all of the beautiful variation that we see in people. And when I say that's how we get all of the beautiful variation we see in people, I mean not just in their physical appearance, but in their behavior, which is a result of their brain. And your brain, the blueprints for building your brain are in your genes. The last thing I want to talk about in this part are X-linked traits or sex-linked traits. And these traits turn out this way because the X and the Y chromosome are very different from one another. When a woman is making an egg, she has to pass on one of her X chromosomes. She has 2 X chromosomes. Most genetically female humans have 2 X sex chromosomes. They are the same size, and they have all of the same loci. So there's a locus on X chromosome A that matches up with every locus on X chromosome B. Make sense? But not for genetically male people. Okay, genetically male humans usually, for their sex chromosomes, have 1 X and 1 Y chromosome. And the Y chromosome is very small. It does important things, like it has the instructions for making testicles and a penis and stuff like that. Stuff that's kind of necessary for reproduction now. But it doesn't have all of the same loci as the X chromosome has. It just doesn't. The X chromosome has lots and lots of loci, and they don't necessarily match up with what's on the Y chromosome. So, for example, color blindness. Let's say that you are a man and you are not colorblind. It means that you got an X chromosome from your mom, because that's all she could give you, that coded for all of the 3 cones. Remember when we were learning about the 3 cones? Okay? So you got an X from your mom that coded for all of the 3 cones. Now your mom, unbeknownst to her, she doesn't know this, but she's actually a carrier for color blindness. She's not colorblind because it's recessive. One of her X's says, don't be colorblind. One of her X's has the code for all 3 cones, and so she makes all 3 cones, and she's not affected. But unbeknownst to her, her other X chromosome is affected. Her other X chromosome has a damaged gene for making the green cone, let's just say. Okay, so she has a damaged gene for making the green cone in her genotype. In her phenotype, you wouldn't know that because her other one is unaffected and is making the green cones. But if dad passes on his Y, you get a son. And if dad passes on his X, you get a daughter. So let's say mom passes on her unaffected X and dad passes on his Y. You will have a son who isn't colorblind. He has the unaffected X from his mom. He has the Y from his dad. So he's fine. You'll also have some daughters. The daughter has to get dad's X. That's what makes her a daughter. And she might get mom's unaffected X. And so she doesn't have color blindness and she's not a carrier. But this daughter gets, yes, dad's X, which makes her a girl. But she gets mom's affected X. That's no problem for her. She's just like her mom. She has one unaffected X that came from her dad. And so now, she's a carrier, but she doesn't know it. However, 25% of the time, dad is going to give his Y and make a son, and mom is going to give her affected X, the one that can't make the green cone. And so that son will be colorblind. And he will have inherited it from his mom. In order for a woman to be colorblind, you have to have a dad who is colorblind. You have to have a dad who has the X chromosome that's affected. And so he passes it on to his daughter. His daughter gets the X, that's what makes her a girl. And mom has to also have at least one affected X chromosome. So she has to get an affected X chromosome from mom and dad in order to be colorblind. And that's why it's very rare for women to be colorblind. In all of my time of teaching classes, I have had one colorblind female student. I've had lots of male colorblind students. Right? One in this class? Ryan, are you colorblind? No? Is somebody colorblind in this class? Is that another class? Oh, you were colorblind. I always think it's cool when someone's colorblind because I get to make an example, you know? Your eyes are normal. What a disappointment. Okay, just kidding. Calico cats are always female. Or, well, they always have two X chromosomes. And here's why. The locus for black fur or dark gray fur is at the same place as the same locus for orange fur. So you have, and it's on the X chromosome. So you have to get two X chromosomes as a cat in order to have one be black and one be orange, or one be gray and one be orange. They're co-dominant, they both express themselves, but they're both on the X chromosome. And so the cat must have two X chromosomes. The only time you have exceptions to this rule is if they also got a Y chromosome, so they have to get two X's plus a Y. And in that case, they're actually genetically intersexed. And that's very, very rare. Like 1 in a million, they guess. I had a friend who had that. She had a cat that had XXY. They were very rich. They got genetic testing on the cat and stuff. And it was a hermaphroditic calico cat. But that's very rare. Okay? If you see a cat in the Humane Society or anywhere else and it has calico fur, it has two X chromosomes. So most of the time it's going to be a girl. Okay. Does anybody have any questions about that? Okay, that's that lecture.